Recall, the goal of mitosis is to make two diploid cells that are genetically identical to each other and genetically identical to the parent cell. In meiosis, the goal is to undergo two cell divisions, ultimately forming four haploid cells. Each will be genetically different because of crossing over and independent assortment of chromosomes that were originally inherited from the parents. So let's see what that process will look like for this cell here. We will start at the beginning of the cell cycle. That would be the G1 phase of interphase. Before we begin, let's concentrate on what's going on with the chromosomes. So what is the total number of chromosomes in this cell? Notice that there are 4 chromosomes, and each chromosome is just made up of one strand. It is not made up of 2 sister chromatids. Note this is a diploid cell because there are 2 sets of chromosomes. There is the set in blue that was originally inherited from dad, and the set in white that was originally inherited from mom. Between the 2 sets there are homologous chromosomes, so these 2 short ones represent a homologous pair of chromosomes, and these represent a homologous pair of chromosomes. The 2 long ones do. The G1 phase is the period of growth and development of eukaryotic cells. Important restriction checkpoints must be met before the cell can enter the next phase of interphase, the S phase. The S phase, or synthesis phase, is a phase in which DNA replication happens. The 2 strands of DNA unzip, and each strand acts as a template for the construction of a new strand, where every guanine base pairs with cytosine and every thymine base pairs with adenine. It's difficult to do that process with my demo, so we're just going to unfold our chromosomes, and each is going to act as a template for the construction of a new strand there. So we need to make exact copies of all of our genes here on our chromosomes. So we'll do that for all 4 of our chromosomes. We also need to unzip them, and in order to do that, we're going to cut each of our chromosomes in half. We are cutting each of the products of DNA replication in half here. The tape represents centromeres, the point at which the 2 molecules of DNA are held together. So by the end of DNA replication at the S phase, how many chromosomes are there? There are 4 chromosomes. Each chromosome is made up of 2 sister chromatids that are genetically identical to one another because of the process of DNA replication. There are still homologous chromosomes, so this represents a pair of homologous chromosomes, and the little short pair represents a pair of homologous chromosomes. I need to adjust my centromere location here. It's supposed to be identical between the 2 homologous chromosomes. We're now ready to enter the next phase of interphase, the G2 phase. The G2 phase continues growth and development of the cell as proteins and other cellular components needed for mitosis are made. Centrioles needed to construct the spindle apparatus are duplicated as well. By the end of the G2 phase of interphase, the results are similar to that seen in mitosis in that there are 4 chromosomes. Each chromosome is made up of 2 sister chromatids. This is a diploid cell because there are 2 sets, the set that was originally inherited from mom and the set that was originally inherited from dad. Between the 2 sets are homologous chromosomes, similar in size and shape and position of genes along the chromosomes. Prophase 1 of meiosis 1 is a busy time. The centrioles that duplicated during G2 phase of interphase migrate to either side of the cell. The nucleolus disappears. The nuclear envelope breaks down. And note that the chromosomes align with their homologs, forming a tetrad. This close association allows exchange of genetic information between the 2 homologs in a process called crossing over. Crossing over is common. In this demo, I will show a single crossover for each of the tetrads. Recall this is happening during prophase 1 of meiosis 1. And they exchange genetic material. Note that crossing over has resulted in new combinations of genes not seen in either of the original parental chromosomes. Prophase 1 leads to metaphase 1. Microtubules from the spindle apparatus move chromosomes at their centromere until they line up along the metaphase plate. Note the chromosomes are still attached to their homologue and they line up double file. Recall in mitosis metaphase, the chromosomes lined up single file. In anaphase 1, we begin to separate the homologs from one another and pull them to either side of the cell. Remember, in mitosis during anaphase, we were separating sister chromatids. When the chromosomes have reached either side of the cell, we've reached telophase. During telophase, we have the reformation of a nuclear envelope around each of the separated chromosomes, and we have cytokinesis occur, separating the cell into two new cells. Note that following cell division, each of the two new cells only has two chromosomes each. Each chromosome is still made up of two sister chromatids. These are now haploid cells. There's just one set of chromosomes in each cell, one long, one short, one long, one short. Notice that now we have no homologous chromosomes. We separate the homologs during anaphase 1. But note, the sister chromatids are not identical. Due to crossing over, we have new combinations of genes. Now each of these two cells will go through the process of meiosis 2. Note there is no interface between meiosis 1 and meiosis 2. We will see that meiosis 2 is similar to mitosis in that we will separate sister chromatids from one another. In prophase 2, chromosomes supercoil and become visible under the microscope. The nucleoli goes away and the nuclear envelope breaks down. Centrioles move to either side of the cell and radiating microtubules from the spindle apparatus attached at the centromeres of the chromosomes and move them till they line up single file at the metaphase plate, similar to how they lined up during mitosis, making it easy to separate the sister chromatids from one another during anaphase 2. These sister chromatids move to either side of the cell in both of the cells till they reach either pole of the cell, leading to telophase 2 and then to the second cytokinesis. The result is going to be 4 new cells. We have new nuclear envelopes forming around the chromosomes in each of the 4 cells as cytokinesis separates those cells. We have 4 cells. Here are the 4 cells that result from meiosis. Each cell contains 2 chromosomes each. Each chromosome is made up of one unit. They are not made of sister chromatids. These cells are haploid. They just have one set of chromosomes, one long, one short, one long, one short, one long, one short. Because these cells are haploid, there are no homologous chromosomes. We separate the homologs from one another during anaphase 1. Also note that each cell is genetically different from one another for 2 reasons. One, from crossing over between the homologs during prophase 1, and the second was the random independent assortment of the parental chromosomes that happened during metaphase 1. This will lead to the genetic diversity in the resulting gametes, sperm and egg.